Yes, sir. Please start, sir. We are live now.
All right. Good evening, everyone. Welcome to all the MOOC students and participants. Welcome to the second live session of MOOC courses Bioengineering and Interactomics. We are going to talk to you today live from our lab. Few applications which are based on mass spectrometry based proteomics. What we thought, mass spectrometers have made a huge impact in various type of application. You think about cancer, you think about infectious diseases. Everywhere you will see that, in which way these recent technologies have made huge impact, and they have started identifying some novel targets, understanding disease mechanism, and in many cases, even potential biomarkers. So what we thought today, given that mass spectrometers are very large topics, and in the course you're already studying about the nut and bolts of the different technologies, especially the proteomics and mass spectrometry, we thought to give you the flavor of these technologies directly from the lab by taking one case study on COVID-19. Of course, COVID-19 does not require any more introduction. And we have learned a lot from, during the pandemic. But what was also interesting, in which way our powerful omics technologies, can they deliver at the time when it was required? And that's where we thought, can we use various type of mass spectrometry technology to address different type of questions in the during the pandemic for COVID-19? One thing was, can we take the complex samples like plasma sample from the human which is infected with SARS-CoV-2 and look at their host responses, in which way due to the viral infection, what kind of host immune response and host proteins get triggered. Likewise, we were also curious to see, like, the nasopharyngeal swab sample, which is taken for the real-time PCR diagnosis part. Can we take the same sample and look at, are we able to detect the viral peptides? And can we also look at what are the human respiratory specimen can reveal any host response of human as well? So again, looking at deep host proteome profile from the plasma or swab sample could provide us some novel insight. And likewise, from the swab sample, nasopharyngeal or otopharyngeal swab, can we get the viral diagnosis done? In which way one could understand the viral host interaction, also looking at various technology like immunoprecipitation, mass spectrometry. And then can we also study the immune response using protein microarrays, another type of interactomics technology. So as you can understand, that different type of questions could be addressed from the same type of biological specimens by using these technologies. So before I come to the actual technology and the some of the demonstrations on the lab, let me just remind you that at the time when we were trying to understand COVID-19, first our team collaborated with many hospitals around in Mumbai and looked at the epidemiology of the Mumbai population. And by now these things are not anymore surprising facts, but at the time we were looking at the big data sets to try to understand which age groups are getting most infected, which type of comorbidities are actually affecting the individuals the most, where we can see more of the severe infection. And again, some of these type of epidemiological analysis helped us to understand that which are the patient group which should be taken more seriously for the investigation and looking at more molecular mechanism and the proteomics investigations. So this work was at that time very relevant, was published in one of the Lancet journal, Clinical Medicine. We then focused on looking at the patient's nasopharyngeal swab sample. And before I move forward on this part, let me first acknowledge different clinicians, Dr. Jayanti Shastri, Dr. Sachi from the Kasturba Hospital and KEM Hospital, and Dr. Om Srivastava from Jaslok Hospital. All these clinicians were very helpful to provide us the sample after all the due diligence of regulatory approvals to start this type of research. And we were very curious. We had not done any type of nasopharyngeal swab proteomics prior to the pandemic. So we had a lot of apprehension that what kind of interference which we may see from these kind of samples, because when you are looking at the swab sample, which is with the VTM, what kind of interference we will have. how to inactivate the virus and also still able to get the viral proteins and peptides. All of these are quite challenging questions at time. So I don't want to make today very elaborate another lecture, but rather just to set the context, I'll go through some of the slides rather quickly to give you a glimpse of what work we have done. But then what we want to spend more time is essentially directly show you these instruments from the lab and take each application one by one with TAs of this course who have been interacting with you, who will directly give you more insight about different technologies in different application context. So as you're aware that mass spectrometry based proteomics has really become powerful with more and more approaches coming with the shotgun proteomics. Now we do not rely too much about separating proteins on the gel, but rather we are directly preparing the lysates, which could be digested, and those digested peptides could be, after cleanup, could be subsequently analyzed for the MS and MSMS analysis, followed by data analysis. Now, data analysis becomes very crucial because in this kind of work, when you are looking at large number of peptides, what type of database you're going to use becomes very important. Just imagine in the case what we are talking today is about nasopharyngeal swab sample from the patients. Think about we are taking from the human respiratory specimen. It means there will be human protein there. And if there is viral infection, then we will have those SARS-CoV-2 peptides we may have as well. And further, if we have any co-infection, any type of other bacterial or fungal infection, even they might reflect in these peptides. So now what we got is a cocktail of peptides from different organisms, from the human, from the virus, and maybe potentially from other organisms. So database search and bioinformatics becomes very critical, as you can understand. So one of the technology which is quite one of the workhorses is the Orbitrap fusion, which we used very actively for separating the complex proteome. And then further, we used different type of quantitative approaches, which includes both label-free and label-based technologies. Orbitrap can do with the tribrid technology with the Quadruple, Orbitrap, and ion trap provides you more flexibility of analyzing the different ways of peptide analysis and gives better quantitative ability. We primarily focus on label-free quantification. So every sample was run individually. And what we wanted to compare that what happens to in each sample on a given retention time, what is the spectral count difference in the peptides which and their relative intensity. Can we use that information for understanding the difference between the mild patient and severe patient with every single protein and every single peptide? So that's the kind of power of these technologies, that you can dissect out all the entire proteome and then try to look at systematically for each protein what kind of fold changes you have. Alternatively, one could also do the label-based strategy. Some of you who may be a little new to proteomics, there are very different ways of doing the quantitative proteomics. This means you want to compare your healthy individual or disease individual, control and treatment. You can do either label-based approach. It means you're introducing some labels which can be used to do a readout eventually, or you can use the label-free approach, which is just I described here. So in label-based approach, the peptides are labeled with the isobaric tags like tandem mass tag or I-track, which is another way of looking at the labeling. So again, both of these I-track and TMT could be working on the isobaric labeling only. And I'm not going to go in detail of principles of these, but idea is that they should not add any extra mass on your given sample. So 3 are mild patient, 3 are severe patient. Then you would like to make sure that they're all isobaric. There is no separate mass added, which makes it different. And then they are going to be co-eluted, analyzed further at the MSMS level to show you what kind of fragmentation we have for reporter ions. And that could be used for the further quantitative analysis. DIA or data independent acquisition is a different approach. Both of these are DDA-based methods, while data independent acquisition looks for acquiring the spectra with much more higher resolution with the small windows of M by Z, and then try to use that information for further, giving you high resolution data, which could be utilized with the spectral library comparison. So again, some of these could be easy for you to follow if you have already take this lecture by now. Some of you who will be taking this in some time in your course, you will be introduced in much more detail. But we just want to apprise you that, when we say proteomics, we had many possibilities to explore, to think about how we can do this analysis. However, the major question was, can we do the good protein extraction? Because we had no immediate that time, the viral safety was a major concern. We did not have the prior experience of handling the swab sample and doing inactivation of virus. So working with these kind of SOPs was very critical. And then, of course, once the protein extractions are done, then one could move on to the looking at mass spectrometers to resolve them better. The broad workflow, which I think I have shown you earlier as well, that we are looking at the leftover VTM sample from the patients, extracting with different type of organic solvent, which will inactivate virus and also extract the protein, digest them, and then either in orbitrap fusion to look at the global peptides, or look at the targeted skyline-based approach, triple quadruple mass spec, to look at selected reaction monitoring for specific peptides. So I'm just showing you this workflow here, or the sample prep. Of course, it will be really difficult to show you all these steps in the very short time. But just to again remind you that sample preparation is the most critical part for any type of high throughput omics-based work, whether you talk about genomics, transcriptomics, or proteomics. In this case, we had to really first come up with, can we take these VTM samples and first inactivate them by doing the heat incubation and making sure that virus is inactivated, because you cannot allow these kind of sample to come in the lab and running the instruments without ensuring the safety of this. And again, then there's a way off that, how much you want these type of sample to be, heat treated. So you have to optimize that. But by that time, we had some WHO guideline and some of the recommendations from other labs, which also helped us to optimize these protocol. But then it was very safe to work with this type of samples with 65 degrees incubation for half an hour or 45 minutes. Then we looked at after extracting the protein, running them on the gel, that they look good. Then we injected in the mass spectrometer after peptide digestion, and we could find that variety of extraction methods from isopropanol, ethanol, or acetone, they're all actually able to give us some unique peptides of SARS-CoV-2, but the pool can give us the maximum peptide, which represents variety of protein from spike protein to nucleoprotein, replicase polyprotein. So then we thought, can we take the cocktail of these picture and use that for further investigation? Again, as I mentioned to you, you can look at variety of proteome comparison from the this type of sample. And we thought, can we look at the first host response? What is the human protein changes happening because of the viral infection? So any type of comparison when you do, you need to make sure that your instruments are reproducible, your data quality is reproducible, and that's where we run same complex samples over the time so that we can see that they are actually reproducible. Like every sample, when you're running 3 or 4 patient sample, after that you run one QC pool sample, which should be done throughout that several weeks or month time while analyzing the data, so that you have a reference to compare that your instrument quality and setup was reproducible, which looked pretty good in this case. After comparing the host responses, we could see there are these clusters are forming, which is the red one is a positive group and the blue one is a negative group, which was expected that you will have 2 separate groups of the negative COVID, negative and positive patients. While the violet one, which was a bit of surprise, these are the recovered patient. So in some way, these are negative COVID-19 patient, but they also were mapped pretty much like the positive. And while looking at the patient history and talking to clinician, it turned out to be that they had just recovered patient. It means they were still probably carrying the trace of viral infection, which was not detected with the RT-PCR, but probably could still be seen at the peptide level from the mass spectrometers. So that showed you that why mass spectrometers can be more sensitive. And looking at the data, one could actually then segregate the patients which are from the mild group of or the non-severe group versus the severe complicated group of the patients. So several proteins turned out to be pretty different in the 2 groups. But what we also first looked at, can we look at the clinically relevant proteins like lactate dehydrogenase protein, various hemoglobin protein, STAT protein. Some of these were already being used in the clinical setting. But of course, then we thought, can we look at much more meaningful biological sample, which is collected longitudinally over the different time period, which is a plasma sample for the patient. Because if you want to monitor the biochemical parameters, various type of to understand that, how the patient is responding to the drug or how patient is getting deteriorated, various type of clinical tests being done on the plasma. So leftover plasma sample could be very powerful to have the patients continuously being monitored. But when you don't want to take the swab sample after 3 days, 5 days, 7 days, 10 days, 14 days, rather the plasma sample already being collected. So that could be used for looking at host response better. So we took the plasma sample and we did depletion of the abundant proteins like immunoglobulins and albumin, so that those can be removed. At least partially, they can be depleted, as you can see in the gel, as compared to undepleted here. And then we proceeded with the mass spectrometry workflow. And the top middle panel shows you what I already spoke about, the swab samples. So both of them in different ways are showing us the host response. We had quite a bit expertise of looking at plasma proteomics. And at the same time, when we were working on this project in 2020, we had just published this paper in Nature Communications Biology, which was talking about different type or severity of malaria, looking at cerebral malaria and severe anemia as compared to the mild or the non-severe falciparum malaria. So since we had expertise of doing this work and already we had the reagents available, we thought we can expand the study on 200 or so patients to look into the how we can look at plasma proteome analysis. Again, as the visuals from the sample prep you can understand, more than doing the science, the safety of people, the PPE and all the protection and biosafety labs, all those are very much critical to perform these projects. And then, of course, bringing a sample with the safety transports and all the biosafety level of cleanup, all of those were very much challenging. But the technical side, when you have this plasma sample, you can pass them from a column which can deplete out some of these abundant proteins. And then after a depletion of this protein, then you can do the digestion of these proteins, and then those peptides could be taken forward for further analysis. So again, I'm not going in great detail of the entire protocol or the exact workflows, but giving you the flavor so that shortly you will be ready to have the live lab demonstrations. This is a nutshell of this work of comparing the negative and the positive group. Within positive, we have mild and the severe group. And after comparison of these groups, we could see many proteins which turned out to be very interesting, like angiotensinogen protein, alpha-2 microglobulin, S100A8 protein, apolipoprotein. These were all very much different in non-severe and severe group of patients. They also gave us some idea that what is happening mechanistically inside the patient's body. And this was the swab data, which helped us to understand that neutrophil degranulation pathway and some of the translation machinery are majorly perturbed in these patients. And some of the literature review also were pretty much confirming that what we are finding in the right direction. And similarly, we looked at the patient plasma sample and the host response, and turned out to be complement activation pathway and leukocyte activation pathways were major pathways which are getting changed. We also then did a pilot study on around 25 patients to look at the longitudinal profile. And that was very challenging because you have to find the same patients and their 2 time point sample from the mild and severe group, which we did this longitudinal study. And that also pretty much confirmed that, leukocyte activation, alteration pathways, and vitamin D binding proteins are majorly seen changing in these 2 different time points. And this particular work was published in JPR, which was also in the cover page of the journal. So again, as I said, I don't want to make it another lecture today. You have been watching our lectures every week. Today, now we want to spend more time with you on the lab-based demonstrations and giving you some time on the instruments and experiments. So the next 3 major applications we are going to directly take from the lab with the help of TAs of the course, who are going to introduce you once more and then going to talk to you about these applications. So let me first request to start the quantitative deep proteomic analysis. It means you are looking at all the protein from a given sample and their protein identification. It could be a simple band. From that you want to identify protein, or a complex mixture of the protein and you want to look at all their protein identification. All of these things are possible with the different type of LC-MS based approaches. All right, so are we ready for the live session from the lab? Argyo and Abhilash?
Yes, sir. Yes.
Yes, sir.
You are muted. Please unmute. Argue, you're not audible. Argue, please unmute yourself and then start speaking. We can't hear you.
So this is the sample compartment where the samples are injected. So you can find here the peptide samples which we will be going to explain in the slide the sample preparation process. After the peptide preparation, the peptides are brought here. After the peptide preparation, the samples are brought here. The samples are put here. You go to the nanoanalysis due to fragmentation. It has two kinds of form. So once the sample are being injected, so this is what you are seeing is the mass spectrometer orbiter based mass spectrometry. This is the three column. This is the analytical column. From the three column, the sample passes to the analytical column and it is subjected to the fractionation. So this is the probe from which the fractionation happens. And if you can come little bit close, then you can see the ITC also. So from where the high voltage are maintained. So you can see that silver one is the ITC. From here the high voltages are maintained, and we do monitor it with the help of the camera, like whether the bubbling issues or the various issues are coming on that. And finally, we acquired that kind of chromatogram. So Abhilash will be explaining you about the chromatogram.
Okay, so once we inject the samples, now we want to look how many proteins we can cover, and for that depends how much, I mean, what is the length of the column we are using. So this kind of experiment, the quantitative proteomics of the swab and plasma sample, as discussed by sir. So we first prepared the peptides, and then we run using the orbitrap fusion. And once the run is done, now we are trying to look at the chromatogram to interpret how many proteins we can get. So we will process the chromatogram using a software called Proteome Discoverer or MaxQuant, where we will get the number of proteins and the amount of proteins, right? So how that proteins the software predict that is important. So, but before going that, we will generally see how the chromatogram looks and what is the fragmentation from each the chromatogram. So suppose this is one of the fragments, one peptide. Then how many fragments did this peptide come? So for each MS, we will check the MS fragments, and on the basis of this MS fragments, we will. So these all are single unique MS, and on the basis of this, we will identify as well as quantify the peptides, and then using different tools from the peptides, we will convert it into proteins. And then the abundance can be calculated using different statistical tools. Okay, so each and every peptides will generally predict and generally quantify, and after quantification we do the basic analysis. So you can see a sample is running now. This is how we expect a chromatogram to come. So this is a 2-hour gradient we generally run for swab and plasma samples. That is getting run now. So it ran till I think around 52 minutes. So it will run till 120. But the most important while running a sample is looking at the methods. So whenever we run samples, we first optimize the method. So we'll just give a demo how the method looks. So since we are doing LC-MS, so while running samples, we look at both LC parameters and the MS parameters. In LC, how much volume of the sample we are picking? What is the gradient? The gradient is a much needed thing. And on the basis of gradient, we expect the samples to be eluted, right? And then we just do a brief equilibration of column and three columns. And after that, we go to the mass spectrometry. So in mass spectrometry, we try to kind of identify and resolve each and every precursor as well as the product ions. where we give a setting of kind of, in the orbit wrap, the resolution we set around 60,000 with a scan range of 375 to 1700, and then different parameters we optimize. Similarly, the fragmentation also we need to set, where we can show this DBMS2, okay, and various parameters like the HCD, that is the collision energy, how much energy or gases we are supplying to fragment or to break the precursor ions. Those things are important. So we place different parameters to optimize the methods, and whichever method suits best, then we finalize it. So likewise, we have optimized the method for this, and accordingly, this is one of the samples that is running now. So basically, after running this, we will go to different softwares to interpret the data.
Thank you, Abhilash.
Right?
And
so this
Arjun.
was the deep proteomics. And the similar thing we can do if we have a pure protein or some unknown protein. So suppose if we run a sample using a gel.
Yeah, can I just interrupt you for Abhilash? Yeah.
Okay.
All right. Sorry. Because of several applications we have to interrupt this particular part. So idea is that irrespective of what sample you have taken, if you have a small gel piece, which is a more simpler sample, whereas if you have the directly prepared the sample, which is the complex lysate, you have much complex sample. But those samples you can directly take from the LC-MSML based configuration. From the LC, you want to elute out the peptides. If you have these simple band or spot, you can run a short 30-minute gradient. If you have the complex sample, you can run maybe 2 hours or 3 hours long, and then you can get the coverage. So this was the deep proteome analysis using one of the most advanced and high resolution mass spectrometer, orbital fusion. Let me now request Surabhi from our team to talk to you about another application. If you were to do immunoprecipitation to look at interaction analysis, and that was also very important to really know that in the beginning of the pandemic, that this viral protein interacts with which other protein, and what is the viral protein and host proteome interaction happens. So that is next application which will be talked by Surabhi.
Hi,
yeah.
I hope my screen is visible.
Yes, go ahead.
So hello everyone, I'm going to talk about this application of mass spectrometry based proteomics, that is Interactomics. Interactomics, as the name would suggest, look at the interactions of the proteins. So for talking about SARS-CoV-2 and its interactome with the host proteins, this is the experimental workflow for experimental workflow to identify what all proteins the viral proteins are interacting with. Now, this is called Affinity Purification Mass Spectrometry. The workflow includes expressing viral proteins along with an affinity tag in some cell line model, and using the affinity tag to pull down that protein and the interacting proteins along with the viral proteins, and followed by identification of the interacting proteins using mass spectrometry and further pathway analysis. So similar on this note, this work was done by many groups during the pandemic. And this is a SARS-CoV-2 viral host protein-protein interaction network, which using the similar experimental workflow was used to identify various cellular proteins interacting with SARS-CoV-2 proteins, and plasmids encoding all the viral genes with a tag that called the 3 flag epitope were expressed in this HEK293 cell line, and the interacting proteins were co-purified. Another study by this group, Gordon et al., was done, and they also identified various proteins interacting with the viral proteins. And just as an example, this viral protein ORF9b, which was identified to be interacting with this host protein TOM70, as you can see on the screen. This particular interaction was validated in another study. So they proved that they showed that the ORF9b significantly reduced this expression of a particular protein, interferon, and the expression of this host protein TOM70, it largely rescued the overexpression of this interferon, and this was through ORF9b mediated inhibition. So they sort of also validated this interaction that was identified in Interactomics study. Another variation of Interactomics is co-immunoprecipitation. And currently we are using this particular workflow in our lab to identify autoantibody targets in COVID-19 patients. Autoantibodies are antibodies which are directed against the self, that is the host proteins. So using a particular protein that has affinity for IgG, that is the IgG immunoglobulins, this particular protein G, has affinity for IgG. So using a matrix which is attached to this protein G, we are using this to pull down IgGs from the patient samples. And here you can see a schematic representation of that. So the plasma has all the immunoglobulins and antigens. So we are using this protein G to pull down IgGs, and with the IgGs, the host proteins will also be pulled down. And then we run it on gel and see the profile. For example, this one is how the profile looked like. And after subjecting them to LC-MS analysis, we can identify the proteins that were interacting with the IgGs. So this way, co-immunoprecipitation coupled with mass spectrometry can be used to investigate autoimmune response and identify antibody targets in serum samples. Thank you.
All right. Okay, so let's continue with more of the applications. I hope the students and participants who are watching this session can see that these technologies can be useful for so many applications, right? We are just talking about one by one that how the same instruments can be used for deep proteome analysis or looking at various type of applications. And now we talked about immunoprecipitation. Let's next talk about metabolomics. Let's just change the flavor from proteomics, because the same instruments could also be used for the metabolite analysis. And again, at the time of pandemic, we thought we are taking the serum or plasma sample from the patient. Why not we try to look at which are the small molecules or metabolites are also getting changed in these patients' sample. So Shalini from my lab, and who's also a TA for the bioengineering course, she actually did this particular work along with other students from the lab to look at the metabolomics profiling of COVID-19 patient. Idea was to take the mild and the severe group of patients, compare which are all metabolites are getting changed in these two groups when we compare them, and can we identify a set of metabolites which could be differentially regulated, like what we have seen in the proteomics. So let me now request Shalini to talk to you broadly about metabolomics and show some workflow in the quantitative mass spectrometer for this particular application.
Thank you, sir. So I hope my screen is visible. So yes. Hi all, this is the basic workflow. So as sir has already explained that all the mass spectrometers can be used for different applications. So one of the applications is metabolomics, because metabolites are basic basic structure or basic regulators of all the pathways. So here what we tried to do is that we tried to take the nasopharyngeal swab was collected in the VTM tubes, and also we had collected the whole blood samples, which were from which the plasma was isolated. But again, these samples are very crucial, and then these samples have active viral, it can have active viral load. That's why the first step, the first crucial step is to inactivate the viruses. So we have multiple methods to inactivate the viruses. One of those which we use is by using the ethanol method. So the two volume of the ethanol was taken for the serum sample, and it was incubated for 5 minutes at room temperature. Then the ethanol was allowed to evaporate in the lab so that nothing is coming out can affect the people working on the samples. And then after that, when the virus was inactive, then after that it was taken for extraction of the metabolites. This metabolite was extracted by using the methanol in 4 volume of the sample, and then it was kept for incubation for overnight at -20 degrees Celsius. Then the centrifugation was done so that the protein content is pellet down, and whatever remains in the supernatant is the metabolites. Then the metabolites were concentrated using the vacuum concentrator, because when we are going through multiple steps here by adding ethanol and methanol, then we are diluting the samples. Already the metabolites in the serum sample or the plasma sample is low. So we need to concentrate the metabolites what we have extracted so that the profile of these metabolites can be seen and evaluated properly. Then these are added to the glass vials along with the internal standard, which is we are using to monitor the instrument run every time what we are running, because metabolites are highly dynamic. So we need to have internal standard to check whether the instrument is working properly or not, or the variations are whether the variations are coming because of the instrument or because of the biological variation. Then the LCMS was done. So this part will be shown by Avinash and Nirjar shortly. So after that, after the run is complete, the raw files are taken up in the compound discoverer software, and then it is analyzed for each peak, and each peak is annotated by its MSMS pattern. And then once these things are obtained, the Excel sheet in the form of P cross N matrix is obtained, and then it is further analyzed for secondary analysis by using metaboanalysis, or further exosomes can also be studied. So exosome is a part where we see for the exposures happening due to environment. So after analysis, we were able to see that these are the different pathways which were getting enriched by the significant metabolites obtained. So once we further studied these, we were able to see that these are the major components, like creatinine was involved in the alteration of threonine catabolism, aspartate and asparagine metabolism, and glutamate and glutamine metabolism, whereas propionyl carnitine was involved in fatty acid metabolism, and the oxalic acid and glycine conjugate was involved in triglyceride metabolism. So now I would like to request Avinash and Nirjar to show the instrument and how we had used MS for analyzing the metabolomics part.
Yeah.
So, hi everyone. So, just like any other mass specs, we do have the LC. So, this can be connected both to a nano LC. Nano LC is generally used for proteomics. We have already seen in case of fusion how it works and all. So, now at this moment, we are doing metabolomics with the QEQ extractive instrument. So, we have connected a UHPLC, which is the Thermos Vanquish. So, it has a UHPLC column, which looks like this. It has inside this. And also, as according to any UHPLC, it has a much broader autosampler trace where we can keep our samples. From the column, it goes inside the mass spec. So as any other mass spec, it has an source and it resolves. So this particular mass spec Q Exactive has two mass analyzers, the quadruple as well as the orbit trap with itself. So after each sample is run, so during the run, we can monitor each sample runs like this in the real-time manner, and we can line up samples like this. So after the runs are over, we can qualitatively view it, the base peaks, as well as we do keep some internal standards. Like in this case, we have one internal standard that comes around this RT. So we can check it. Also, after obtaining this raw files, we can go and analyze it in softwares like Compound Discoverer, similar to the Proteome Discoverer in case of Proteomics, and many other similar kind of softwares, and further tertiary analysis.
All right, and would you like to show a little bit whether Pine or any kind of, the QC standards on the...
So this is the standard that I was talking about, the internal standard, result Pine. So its molecular weight M by Z is around 609, as you can see here. And according to our gradient, it comes around 9.57 RT. So, and if someone else also has some other internal standards, they can also pull up the ranges part and add if any other standard they have to put the M by Z value in this range. And once done, okay, it will show the particular where that standard is coming out, eluting out.
All right. Thank you, Rajer, for giving the demo. And thanks, Shalini, for the overview. Again, to all the students, as we have not talked in great detail about metabolomics, that was out of the scope of the course. But given that we are using the same instrument, as you can see, same LCM, SMS being used for proteomics and for metabolomics, it's just matter of using different columns and different solvent system. And you can obtain some very novel information about which are the metabolites, which are also getting perturbed in the host. One could try to obtain some novel information. However, it is not as simple as just having an instrument and you run a sample and you get the data. Metabolomics, while the extraction methods are very simple, just methanol extraction works the best. But looking at a variety of possibilities for metabolite, you have to choose the right type of column. C18 column, helic column, various type of columns can be used. Likewise, you will get large number of metabolites, which is their M by Z values from the mass spectrometer. But their identification becomes very challenging because most of the time you may not find the right type of, the correct matches for those. So you will have to rely on a lot of data analysis, which can give you idea that how many metabolites can be identified. So again, identification with the database is still one of the challenge. Just think about in case of SARS-CoV-2, patients were given several medication. Now, if you take the plasma sample and you look at their metabolites, is that something because of the drug, what we see the metabolite changes, or is it because of the host response to these metabolites, what we see? So again, those kind of things becomes critical to analyze and look into that context, that what we are looking at is the directly metabolite changes, or are they something based on directly the drug which has been given to the patient. So broadly, I hope you are convinced by now that these technologies have the potential to look at the complex proteome and metabolome using these mass spectrometers and give us the, for very unknown samples, give us sort of information in a very short time. Data analysis and databases, of course, remains one of the most challenging aspects for any type of complex data analysis. And that was the challenge in this time as well. We had to come up with the SARS-CoV-2 databases for the proteome search and look at all possibilities for the metabolite search. Nevertheless, once you have identified a set of proteins and metabolites, now you want to validate them. How can you do that? So now, if you were to look at the protein-based validation, if I ask you a question, what kind of technology comes to your mind? You will think something which is in light of antibody-based work, right? Some sort of Western blot, ELISA, other type of immunofluorescence-based assays. So various type of immunoassays, which are all linked to different antibodies, are being used for protein-based test and protein validation. However, just think about in this case, how quickly we can get the antibodies against the SARS-CoV-2, it was not possible for very spike protein, nuclear protein, and other replicases. Can you get the antibody generated very fast? Many of the host proteins are changing. Can we get antibodies very quickly? Probably not. Only whatever is available in the market, that's what can be used. Alternatively, can we use mass spectrometry as a way for even validating our targets? So that's where we are now going to introduce you to next type of mass spectrometer, which is a triple quadruple mass spectrometer, and that is being used for targeted proteomics workflow. Here I'm showing you two different type of mass spectrometers. One is the triple quadruple mass spectrometer, and other is Orbitrap instrument, which you just seen just now, the Orbitrap QE, which we talked to you for the metabolomics, and the fusion, which we talked for the proteomics applications. So idea is that, once you know which proteins are really differentially expressed in your non-severe and severe group of patients, or your control and treated group, or the negative and positive group, you want to validate only those selected peptides and proteins. You don't want to look at thousands of proteins at the same time now. So you are looking at very specific targeted assays. And that's where an alternative to antibody approaches are SRM, or selected reaction monitoring based, mass spectrometry based assays now. So the same actually setup and same configuration and same concept could also be used for the targeted metabolomics assays. Although what I'm going to talk and show you is more in the context and the light of the data that we have from targeted proteomics, but even the same instrument and same workflows are also being used for targeted metabolomics. So again, think about these technologies can be employed irrespective of which type of projects you're going to work on and what is your validation which you want to deliver. So the top panel shows you triple quadruple mass spec, where you have two set of quadruples, Q1 and Q3, which are the four metallic rods, where these peptides, which are ionized form, are going to revolve. And further, to select the most intense ions, you are going to have the collision cell, which is misnomer Q2 is being used. And so let's say one of the most intense orange ion from the peptide of our interest is getting fragmented, and that you are going to further monitor, and you are looking at their ion intensity. And what you're generating is time versus ion intensity plot, and you're looking at these transitions from the the parent ions to the fragmented ions. And then you are looking at how best you can look at from the same peptides, multiple transitions to be generated and to be analyzed to look at their cumulative intensities, which could indirectly indicate you the protein quantification or protein intensity. So this is the broad concept, and it requires a lot of computational work. It requires first to select your peptides of interest, make sure that their length and their uniqueness, all that's being checked in the software on the Skyline. And then you can proceed further to make the transition list, which could be given to the software, and then you can do your data acquisition in the mass spectrometer. Alternative thought is to do PRM, or parallel reaction monitoring. Can we come up with an approach where we can look at all the peptides rather than relying only on few selected peptides? Can we rely on all the peptides and look at their validation? So therefore, the PRM-based approach is being used with the Orbitrap configuration, where you have the quadruple and Orbitrap. And quadruple works as a mass filter, and then Orbitrap, given the high resolution and the high efficiency, it can take all of these ions and look for their further fragmentation. So again, the similar configuration, like collision cell is there, and you got two mass analyzers like Q1 and Orbitrap, which can be used for doing PRM. Similar kind of approach, which is like PRM, is known as SWAT, which is from other technology vendor, which is from the SciEx, which is a SWAT technology, which is also with a triple top, you can do the similar application like PRM, and DIE kind of analysis can be done, data independent acquisition. So there are many approaches, and which is actually gives you more possibilities to explore these workflows, right? So when we look at the this particular project for the SARS-CoV-2 COVID-19, our first goal is to always ensure that our data quality and instrument reproducibility is good. So we run some standard sample, and in this case, the BSA, few peptides are being monitored every day to ensure that we are looking at the good intensity, and we are looking at good performance of the equipment, retention time is reproducible. Once we did that, then we moved on to some of the marker which we identified from the swab sample, which were also clinically relevant. So if you recall that, the first pandemic, 2020, that time it was a very unknown disease where whatever new information we can get, it was very pretty unique. So Some paper reported that, okay, interleukin 6 can be indicative of difference in negative and positive and mild and severe, and doctors started testing that everywhere. Some people reported lactate dehydrogenase may also be a good indicator, or C-reactive protein for that matter, or aspartate transferase for that matter. Again, all of these became individual tests to assess that the quality of the what is going on in these individuals. We thought, can we look at with the mass spectrometers, with our target assays, all of these markers, like, what is happening to these which are clinically relevant? Can we first of all detect them? And very happily, we can detect all of them, right? But more importantly, using our mass spec based target assays, you can look at even much more number of peptides. If you're not relying on one test with one ELISA kit or one biochemical assay, you can actually look at here maybe 30, 40 peptides and different proteins. So now we looked at even multiple proteins, angiotensinogen, apolipo B protein, fibrin gamma chain protein, serpin proteins. All of these proteins which we try to compare their non-severe and severe response. And that was pretty dramatic. Like, we could see a huge difference along with the clinically relevant marker, which we can also test on the same sample. Now we can also look at what is happening for the newer protein, which nobody was able to test out with other assays. So that gives you the good number of protein as a way to indirectly assess the patient's possibility of moving from the mild to the severe form of disease. And that became very important because now you have a set of protein which may indicate how a patient is actually deteriorating and probably can be taken forward for the ICU or more kind of therapeutic interventions. Throughout of this process, you have to make sure that you are spiking in some heavy labeled synthetic peptide in every sample, and that should show you the same intensity and same retention time, because that should not change whether you are running the mild sample or severe sample, right? So again, we do want to keep giving you idea that while doing the experiments, you have to keep lot of controls and lot of standards in mind. And based on this type of assays, even we were able to look at viral peptides and look at set of 11 peptides from spike protein, nuclear protein, and replicase polyproteins. In 10-minute solution, can we look at their profile to compare their intensity values and also correlate them with the CT values. So the last demonstration is going to be now on the targeted proteomics with our team, and probably Avinash is going to take you through the targeted proteomics workflow. And Medha is also there. Yes, Medha, go ahead.
Hello everyone. So I'm Medha. I'll be talking about the instrument. I'll be showing you the instrument here. So here we are connected an HPLC to our triple quadruple KSQ Altus. So this is where we place the samples. You can see we have placed the samples here. The vials are here. The injection happens from here. The sample will be taken up. As you can see on the screen, one sample is running right now. This is our standard QC sample. This is the triple quadruple mass spectrometer on which we do the targeted assays. So right now we are running BSA to check the daily instrument output, how it is coming, how the response is coming. So you can see it is a 5-minute gradient. You can optimize the gradients, and according to how sir has said, depending on your samples, you can vary the gradients, optimize the transition list, and all that can be done. So I will show you one of the runs that were done earlier. So for example, we run daily some complex samples and we monitor the response that is coming. As you can see, this is a standard cell lysate. Here a few of the proteins we are monitoring, and we can see how the day-wise response is changing. So for 3 consecutive days, you can see here the response is more or less similar. This day was, we have kept as reference, which was way before the these 3 consecutive days. You can see the response is same. Even similar for another standard sample, which is BSA, a purified protein. Here also you can see we monitor certain proteins for which we have refined the method, and this is how the peaks look like. They are all similar looking, and the response is almost equal. So this is the software with which we operate the LC, the HPLC unit. Here you can see we can operate the pump, the sampler, and the oven. In the sampler, we put the information of where the vial is kept. In the pump, we get the information of how much pressure is there for a particular column and how much flow rate we have kept. And this is the column related tab. Here we will put details about what temperature the column should be at while the run is going on. So currently, since the run is going on, it will keep changing based on the method that we have optimized. Now the sample acquisition will start. Here you can see the sample will be injected. The sample is being injected here. So now the injection needle will move to the vial. So it is moving to the vial and now it will take up the sample. After this, it will inject the sample into the column. And from the column, you can see the column is here. And from the column, the line goes to the mass spec. And we will get the response here.
All right. Thank you very much, Mehta, for doing this demo for targeted proteomics workflow. We are almost at the end of the session now. So first of all, let me thank all the TAs for doing the perfect job at the very defined time which we wanted to show you demo. And as you can see, it is a lot of coordination required among so many people and on different instruments to really meet everything within that 1 hour time assigned for this live session. I hope the participants are able to follow these things that, beyond the theory which we talk in the course, how in the real life, how some of these problems which we would like to address, especially looking at the clinical proteomics and metabolomics, could be addressed by using these technologies. And mass spectrometry has really become one of the very powerful resource and the workhorse in the omics technologies. Before we close, let me announce you once more and remind you that for the bioengineering course, in addition to all of our TAs who are following up your forum and interacting with you, we also have a live session every Friday with Lavanya, who is talking to you about some assignment. Do interact and try to take some of this opportunity for further questions. We are also having a bioinformatics live session for both the groups, bioengineering and interactomics group, which Ankit is taking for introducing you to some new tools of bioinformatics, just much beyond than just the course and the topic which we're talking, to give you a better feel of how you can do some of the things at your home and in your place yourself. So I hope you are enjoying both of these MOOC courses, bioengineering and interactomics. You had several questions which we thought we'll talk, but given the short time, let me just take one question which I recall was essentially talking about the role of mass spectrometry and how one could actually think about a future career possibility in this area. So just let me mention once more that if you are currently getting trained in the technologies like mass spectrometry, and if you can do proteomics or metabolomics or these applications, you are going to be in great demand because of the way these technologies are so powerful and the way many applications can be done. There's a lot of focus from the pharma sector and various type of R&D labs to really utilize the mass spectrometer for various type of applications. So your basic theoretical knowledge and your practical experience can really bring you to a good market for the jobs as well, along with learning what you're learning in the current MOOC courses. So we'll be happy to interact with you more and give you more flavor of these technologies. And at the end of courses, the students who are really topper of the course, we usually give them opportunity to come to our lab and do some short internship as well. So that, again, if you're performing well, you will have more opportunities to interact directly and work in our lab as well. That's pretty much for the today's live session. I hope you have enjoyed learning some of the complex technologies. And we'll again once more interact with you the next month with some new flavor of new technologies. By that time, it will be more about interaction analysis with the more recent approaches of microarrays and surface plasma resonance and biolayer interferometry technologies. Till then, good luck and keep studying and watching our MOOC lectures. Thank you.